Imagine a planet with no living thing on it. Not a single bacterium, not a single virus, not one cell. Just rock, water, and poisonous air. That was Earth 4 billion years ago. And somewhere in that dead hell, in the mud, in boiling water, under lightning strikes, something happened. Something so improbable that scientists still argue about it in hushed, almost reverent tones. Dead chemistry came alive. Before we continue, please subscribe to this channel. Your support allows us to continue telling our story. Thank you. Now, let's go back. All the way back. Not to the beginning of human history, not to the dinosaurs, not even to the first fish. We're going back to a time so remote, so alien, that the very ground beneath your feet did not yet exist in any form you would recognize. We're going back 4,300,000,000 years, to a planet that had just survived something catastrophic. A period scientists call the Late Heavy Bombardment, when the inner solar system was a shooting gallery and Earth was being struck by asteroids and comets on a scale that is almost impossible to picture. The sky was not blue. It was orange and brown, thick with carbon dioxide, methane, ammonia, and water vapor. There was no free oxygen, not a single molecule of it. If you could somehow stand on that surface, you would be dead in seconds. Not from the heat, though that was considerable. Not from the lack of oxygen, though that would finish you quickly enough. You would die because everything about that environment was hostile to the kind of life that eventually learned to walk upright and ask questions about its own origins. And yet, from that environment, from that hellscape of toxic gases and boiling shallow seas and constant geological violence, something emerged that would eventually produce every living thing that has ever existed on this planet. Every tree, every insect, every whale, every bacterium in your gut right now, quietly doing its work. Every human being who has ever drawn breath. All of it traces back to a single moment, or perhaps a series of moments on that poisonous, beautiful, catastrophically violent early Earth. The question is not simply, how did life begin? That question, as simple as it sounds, is one of the deepest and most contested in all of science. It sits at the intersection of chemistry, geology, physics, astronomy, and philosophy. It touches on questions about what life actually is, which, as we'll discover, is far less obvious than it seems. And it has attracted some of the most brilliant, most obsessive, and occasionally most eccentric scientific minds of the last 2 centuries. Here is what makes this story so extraordinary. We are not talking about some distant, abstract event. We are talking about something that happened to the atoms that are currently inside your body. The carbon in your cells was forged in dying stars. The hydrogen bonded to your DNA was present at the birth of the universe. And somewhere along the way, through a chain of chemical accidents and improbabilities so long and so intricate that it staggers the imagination, those atoms organized themselves into something that could grow, respond, reproduce, and eventually, billions of years later, wonder where it came from. That is the story we're telling tonight. Let's begin with the planet itself. 4 billion years ago, Earth was roughly 100 million years old, young by cosmic standards. The Moon had only recently formed, and the leading theory for how that happened is itself extraordinary. A body roughly the size of Mars, which scientists have named Theia, struck the proto-Earth in a collision of almost incomprehensible violence. The impact was not a gentle, glancing blow. It was a catastrophic merger that melted both objects, threw an enormous cloud of vaporized rock into orbit, and eventually coalesced into the Moon you see in the night sky. That Moon, at the time, was much closer to Earth, perhaps 10 times closer than it is today. The tides it generated were enormous, washing hundreds of miles inland, churning the coastal shallows into a constant, chaotic mix. The early ocean was everywhere. Not quite as salty as today's ocean, but acidic in places, alkaline in others, warm in shallow areas near volcanic vents, cold in the deeper basins. The ocean covered most of the surface, interrupted only by volcanic islands and small, unstable land masses that were constantly being rearranged by a tectonically hyperactive planet. The mantle was hotter. Volcanoes were more numerous. Hydrothermal vents pumped minerals and heat into the water. And the water itself was rich, not with life, but with chemistry. With the raw ingredients that, under the right conditions, might begin to do something remarkable. Now, pause here for a moment and consider the bombardment itself more carefully. Because it was not simply a catastrophe. It was, in the most literal sense, a delivery. The asteroids and comets that struck early Earth were not made of bare rock. Many of them were carbonaceous chondrites, a class of meteorite so rich in organic compounds that they are essentially flying chemical laboratories. The Murchison meteorite, which fell in Australia in 1969, has yielded more than 70 different amino acids upon analysis. Far more varieties than the 20 that life on Earth uses, and many of them identical to the ones in your proteins right now. They had assembled themselves in the cold chemistry of the early solar system, hundreds of millions of years before Earth existed, and they arrived here as a molecular inheritance. But the comets brought something even more surprising.
In 2014, the European Space Agency's Rosetta mission placed a small lander on the surface of Comet Churyumov-Gerasimenko, a 4-kilometer lump of ice and rock traveling through space at tens of thousands of kilometers per hour. Among the molecules detected on its surface was glycine, one of the simplest amino acids and one of the most fundamental components of protein in every living thing on Earth. Glycine, on a comet, assembled by chemistry alone in the darkness between the planets billions of years before any organism existed to make it. The implication is staggering. The molecules of life did not originate on Earth. They are a cosmic inheritance, a molecular toolkit assembled across billions of years and trillions of kilometers of space, delivered to this planet by the very catastrophe that seemed most likely to prevent life from ever arising. The late heavy bombardment, in other words, was not only a threat, it was also a seeding event. The violence and the gift arrived together. What were those ingredients more precisely? Here is where the story gets genuinely strange, because the building blocks of life are not rare. They are, in fact, embarrassingly common. Carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. The 6 elements that make up the overwhelming bulk of every living organism
are among the most abundant elements in the universe. They are found in meteorites. They are found in interstellar gas clouds. They are found on the surface of comets. When the Murchison meteorite fell in Australia in 1969, scientists analysed it and found something extraordinary. It contained amino acids, the same kinds of amino acids that form the proteins in your body. They had formed naturally in space without any living thing to guide the process. This was not an anomaly. Subsequent analysis of other meteorites found the same thing. The chemistry that produces the molecular building blocks of life does not require life to get started. It requires only the right atoms, the right energy source, and time. And the early Earth had all three in spectacular abundance. But let's be precise about what we mean by building blocks, because there's a temptation to think of this like assembling furniture. If you have all the parts, you just need to put them together. The reality is vastly more complicated and vastly more interesting. Think of it this way. Imagine you have a pile of lumber, some nails, a few windows, and a roof's worth of shingles. These are the building blocks of a house. But a house is not just a pile of materials arranged randomly. A house has structure. It has internal organization. It has a specific relationship between its parts that makes it function as a house rather than just a pile of debris. The gap between materials and house is enormous. The gap between amino acids and a living cell is even more enormous. A living cell is not just a collection of molecules. It is a machine of staggering complexity. A machine that takes in energy, processes information, maintains its own boundaries, responds to its environment, and reproduces itself with extraordinary precision. A single bacterium, which is the simplest form of cellular life we know, contains thousands of different proteins, a complete copy of its genetic code, a membrane that selectively controls what enters and exits, and a suite of molecular machinery so sophisticated that the best human engineers can only dream of replicating it. And this is the simplest known form of life. How do you get from a handful of amino acids floating in a warm sea to something like that? This is the central mystery. And the story of how science has tried to answer it is one of the great intellectual adventures of the modern era. It begins, in many ways, with a young graduate student named Stanley Miller and his advisor Harold Urey at the University of Chicago in 1952. Miller was 23 years old. Urey had won the Nobel Prize, and together they designed an experiment that was simple enough to fit on a laboratory bench and profound enough to change science forever. The setup was elegant. Miller took a sealed glass apparatus and filled part of it with water, representing the early ocean. He filled another part with a mixture of gases thought to represent the early atmosphere: methane, ammonia, hydrogen, and water vapor. Then he ran electrical sparks through the gas mixture, simulating lightning. And he let it run. After just 1 week, something remarkable happened. The water in the apparatus had turned brown. When Miller analyzed it, he found that it contained amino acids, the molecular building blocks of proteins. Not one or two, several, including glycine and alanine, two of the most common amino acids found in living things. The moment Miller saw those results, he reportedly stood in the laboratory for a long time, without speaking. He was 23 years old. He had just made the stuff of life from scratch, from gases and water and electricity, from nothing more than what the early Earth had in limitless abundance. His advisor, Yuri, a man not given to easy enthusiasm, told colleagues privately that it was among the most significant results he had ever seen. The paper they published ran to barely 2 pages in the journal Science. It may be the most consequential 2 pages in the history of chemistry. What followed in the years after Miller and Urey was a cascade of increasingly remarkable results. In 1960, the Spanish-American chemist Juan Oro took the experiment further. He mixed hydrogen cyanide and ammonia in water, chemicals that would have been abundant on early Earth, and heated the mixture gently. What he got was not a simple amino acid. He got adenine, one of the 4 nucleotide bases that spell out the genetic code in DNA and RNA. One of the 4 molecular letters in which the entire library of life is written. Oro had made a letter of the genetic alphabet from scratch, in a glass flask, from the simplest possible starting materials. The philosophical reverberations of these experiments extended far beyond the laboratory. This was the early 1950s and 60s, the Cold War, the nuclear age, a civilization grappling with its own capacity for annihilation. And here, in a Chicago laboratory, a young scientist had suggested something subversively hopeful: that life was not a cosmic accident requiring divine intervention or an improbable alignment of impossible circumstances. Life was chemistry, ordinary chemistry, inevitable, patient, persistent chemistry. Given the right conditions and enough time, it happened. Not because the universe was designed to produce it, but because the universe simply could not help itself. The reaction was not universally welcoming. Religious communities objected. Some scientists objected too, and as geological evidence accumulated over the following decades, the picture of the early atmosphere became considerably murkier. Many researchers now believe that the early atmosphere was dominated not by methane and ammonia, but by carbon dioxide and nitrogen, gases that are far less reactive under electrical discharge, and that don't produce amino acids nearly as readily. The Miller-Urey atmosphere may have been wrong, but here is the crucial point. It doesn't matter. Because in the decades since 1953, chemists have found that amino acids form readily under many different conditions. In hydrothermal vents, on mineral surfaces, in ice, under ultraviolet radiation, in simulated impact events. The specific pathway Miller demonstrated may not be the one nature used, but the general principle his experiment established, that life's building blocks assemble themselves spontaneously from simple chemistry, has never been seriously challenged. It has only been confirmed again and again by experiments of increasing sophistication. But before we follow the chemistry deeper, before we descend to the ocean floor and enter the darkness where the most radical theories of life's origin were born, we need to stop and confront a question that the Miller-Urey experiment raised but never answered. A question that sounds simple and turns out to be one of the most philosophically destabilizing problems in all of science. What exactly is life? You might think we know. You might think that after centuries of biology, after Watson and Crick, after the sequencing of the human genome, after the discovery of extremophiles living in boiling acid and Antarctic ice, we would have a clean, agreed-upon definition. We don't. There is no universally accepted scientific definition of life. Not one. Ask 10 biologists and you will get 11 answers, at least 2 of which will contradict each other in ways that both parties consider fundamental. NASA, which has a practical reason to care about this question, since it needs to know what to look for on other worlds, defines life as a self-sustaining chemical system capable of Darwinian evolution. It is a useful, working definition, but it has problems. A mule cannot reproduce. Is a mule not alive? A virus cannot sustain itself without a host cell. Is a virus alive? A seed can sit dormant for thousands of years, metabolically inert, and then germinate. Was it alive during those millennia of stillness? A fire consumes fuel, grows, reproduces by spreading to new material, and responds to its environment. Is a fire alive? These are not trick questions. They are genuine problems that reveal something important. Life is not a thing. It is a process. A set of interlocking, mutually reinforcing chemical processes that, taken together, produce the phenomenon we recognize as living. And the reason we struggle to define it precisely is that those processes did not appear all at once. They emerged gradually, one by one, over millions of years, each new capability building on the last, with no clear moment at which the whole system crossed a threshold and became unambiguously alive. This matters enormously for the origin of life question. Because if life is a process rather than a thing, then asking, when did life begin, is a bit like asking, when did a river begin? A river begins as rain. The rain becomes a trickle. The trickle joins others and becomes a stream. The stream deepens and widens. At no point is there a sharp boundary between not river and river. There is only the gradual accumulation of water moving in a direction, carving its own channel through whatever landscape it finds. Life, in this view, began as chemistry. Chemistry that was slightly more organized than the chemistry around it. Chemistry that could persist and then replicate and then improve. The transition from chemistry to biology was not a single event. It was a landscape, and we are still mapping it. Now hold that thought, because the landscape had a geography. And that geography was stranger than anything Miller imagined in his Chicago laboratory. Consider what the early Earth actually looked like from above. If you could have hovered somehow in that poisonous atmosphere and looked down at the surface, you would have seen an ocean. An enormous, dark, iron-rich ocean, reddish in color from dissolved minerals, stretching to every horizon. Small volcanic islands rising here and there, steaming, geologically violent, constantly being reshuffled by the hyperactive tectonics of a young planet. Shallow coastal lagoons where the chemistry of the ocean concentrated and dried and reconcentrated in endless cycles driven by the tides. Those enormous tides generated by a moon 10 times closer than the one that hangs in tonight's sky. And everywhere, energy. Energy in forms and quantities that the modern Earth no longer experiences. Ultraviolet radiation pouring through an atmosphere with no ozone layer to filter it. radiation intense enough to drive chemical reactions that simply don't occur in today's world. Lightning storms of a frequency and violence that would make a modern thunderstorm look like a camera flash. Constant volcanism, pumping sulfur dioxide and hydrogen sulfide and carbon monoxide into the atmosphere and into the ocean. Radioactive decay in the crust at rates 3 or 4 times higher than today, generating heat from within. And the impacts, still ongoing though declining, each one delivering a pulse of energy and chemistry to whatever local environment it struck. In that world, chemistry was not slow. Chemistry was relentless. And the diversity of chemical environments was extraordinary, not just from place to place, but from moment to moment in the same location. A shallow coastal pool might be hot and acidic in the afternoon, cooled and diluted by rain in the evening, concentrated by evaporation the following morning, irradiated by ultraviolet at noon, flushed with fresh ocean water by the next tide. Each cycle was a new experiment. Each experiment ran to completion and began again. Charles Darwin, in that famous letter of 1871, imagined a warm little pond. It is a charming image and perhaps not entirely wrong, but the reality was less tidy.
The reality was thousands of warm little ponds and cold deep basins, and hot mineral springs and alkaline vents, and acidic lagoons, and frozen shorelines, and sun-baked tidal flats, all running chemistry simultaneously, all exploring different corners of the same vast chemical possibility space. The origin of life was not a single experiment. It was a planetary-scale research program, running billions of simultaneous trials for hundreds of millions of years, with no hypothesis to test except the blind thermodynamic imperative to find new ways to dissipate energy. And that framing, life as a thermodynamic inevitability, as nature's way of dissipating energy gradients, is not a metaphor. It is a serious scientific proposition, developed most rigorously by the MIT physicist Jeremy England, whose work in the second decade of this century attracted enormous attention and considerable controversy. England's argument begins with a principle of statistical physics known as the fluctuation theorem. In essence, the theorem says that systems driven by an external energy source, like the sun or a chemical gradient, will tend, over time, to evolve toward configurations that are better at absorbing and dissipating that energy. They don't do this purposefully. They do it because configurations that dissipate energy more effectively are thermodynamically favored. They are more stable, in a deep physical sense, than configurations that dissipate energy less effectively. Now apply that principle to a collection of organic molecules bathed in sunlight on the surface of an early Earth ocean. Random chemistry produces a vast number of possible molecular configurations. Most of them are thermodynamically unremarkable, but occasionally, rarely by chance, a configuration arises that is better than average at absorbing sunlight and converting it into chemical reactions. That configuration is thermodynamically favored. It persists longer than its neighbors. And if it happens to also catalyze its own formation from surrounding molecules, if it is, in however crude a sense, autocatalytic, then it doesn't just persist, it grows. England's work suggests that the emergence of self-replicating chemistry from non-replicating chemistry is not a miracle,
or a statistical impossibility. It is something that physics should lead us to expect, given the right conditions and sufficient time. Life, in this view, is what matter does when it is bathed in energy for long enough. It is a phase of matter, as natural as ice or liquid water or steam, but richer, more complex, more capable of surprise. This idea has its critics. Some physicists argue that England's calculations, while elegant, don't yet make specific enough predictions to be tested. Some biologists worry that it reduces the complexity of life to a thermodynamic tautology. The debate is ongoing and genuinely unresolved. But here is why it matters for our story. Because it represents a fundamental shift in how we frame the question. For most of the 20th century, the origin of life was treated as a problem of improbability, a question of how a sequence of enormously unlikely events could have occurred in the available time. Origin of life researchers spent decades trying to make the chemistry seem more plausible, the time estimates more generous, the probability calculations less frightening. England's approach, and Lane's, and the alkaline vent hypothesis more broadly, inverts the question entirely. Instead of asking, how could something so improbable have happened, they ask, given the physical conditions, how could it not have happened? Instead of treating life as a lucky accident that needs explaining, they treat life as a predictable outcome that needs understanding. This is not a settled question. Science is not a referendum, and the fact that several researchers find this framing compelling does not make it true. But it represents a genuine, substantive shift in the conceptual landscape. And it has implications that extend far beyond the origin of life on Earth. Because if life is thermodynamically favoured, if it is what matter does in the presence of sustained energy gradients, given enough time and chemical diversity, then life is probably not rare. The conditions that prevailed on early Earth were not unique to Earth. Liquid water exists, or has existed, on Mars, on Europa, on Enceladus, almost certainly on dozens of the exoplanets now being catalogued by the James Webb Space Telescope at a rate of dozens per year. Energy gradients, chemical, thermal, radiative, are among the most common features of planetary environments in the universe. If England is right, or even partially right, then the galaxy is not a vast, empty silence punctuated by one improbable spark of life. It is a garden so large that we cannot see its boundaries, tended by no one, requiring no gardener, producing life wherever the thermodynamic conditions are met, which, on cosmic scales, may mean everywhere there is liquid water and a source of energy, and enough time. We do not yet know if this is true. The honest scientist will tell you that we have exactly one data point, one planet where we know life arose. And that one data point cannot tell us whether life is common or rare in the universe. But the chemistry we have explored tonight suggests, with increasing force, that the barriers to life are lower than we once believed. That the path from chemistry to biology, while long and winding and still incompletely mapped, is not a path that requires miracles. It requires only what the universe supplies in abundance: matter, energy, time, and the iron laws of thermodynamics playing out across billions of years. And this is perhaps the most vertiginous implication of the origin of life story. Not that we understand where we came from. We don't, not fully, not yet.
but that we are beginning to understand the kind of story it is. It is not a story about a special planet chosen for a special purpose. It is not a story about a universe that needed life in order to exist. It is a story about a universe so large, so old, so richly stocked with the conditions for chemistry, that life emerges from it the way music emerges from a vibrating string. Not because anyone intended it, not despite the laws of physics, but because of them. Because that is what the laws of physics, given sufficient time and complexity, inevitably produce. Now, armed with that understanding, with that sense of the scale and the logic of what we are dealing with, we can descend. We can leave the surface, leave the lightning and the ultraviolet and the warm coastal pools, and go down into the darkness of the early ocean, where the most radical and perhaps the most compelling theory of life's origin was waiting to be discovered. The question was never whether the building blocks could form. The question was always what happened next. And the answer to that question came from a direction no one expected. Not from the surface of the Earth, but from far beneath it. In 1977, a research submarine called Alvin was exploring the ocean floor near the Galapagos Islands at a depth of roughly 2,400 meters, about 1.5 miles down. The scientists aboard were looking for volcanic activity along the ocean spreading zone, where tectonic plates pull apart and magma wells up from below. What they found instead stopped them cold. Chimneys. Towers of mineral-encrusted rock rising from the sea floor, belching superheated water. Water that had been heated by the volcanic rock below to temperatures exceeding 350 degrees Celsius.
This water was rich in dissolved minerals, iron, sulfur, manganese, and it was creating dense black clouds as it mixed with the cold surrounding seawater, causing the minerals to precipitate out. The scientists called them black smokers. But that wasn't the most astonishing thing. The most astonishing thing was what was living around them. In water that should have been completely inhospitable to life, hot, dark, under crushing pressure, with no sunlight for photosynthesis, there were thriving ecosystems. Tube worms 6 feet long, clams the size of dinner plates, shrimp, crabs, fish, an entire food web operating in complete darkness, powered not by sunlight, but by the chemical energy pouring from those vents. The discovery rewrote the rules of biology. It demonstrated that life did not require sunlight. It did not require the surface conditions that everyone had assumed were essential. It could thrive in environments that, by any prior understanding, should have been completely lifeless. And it raised an electrifying possibility. What if the deep sea vents were not just a curiosity, an extreme environment where life happened to survive against all odds? What if they were the birthplace of life itself? This idea has grown considerably more sophisticated in the decades since Alvin's discovery. The black smokers themselves, those ferociously hot, acidic vents, are now considered less likely as birthplaces, because the conditions are perhaps too extreme, too chaotic, to allow the delicate chemistry of life to get a foothold. But another type of vent
discovered more recently, has become one of the most compelling candidates for life's origin. In 2010, scientists exploring the Atlantic Ocean discovered a remarkable structure they named the Lost City. It was a field of hydrothermal vents unlike anything previously seen. Not hot and acidic like the black smokers, but warm and alkaline. The chimneys at Lost City were not black.
They were white, built not from metal sulfides, but from carbonate minerals, creating ghostly white towers up to 60 meters tall. The water they emitted was warm rather than scalding, perhaps 40 to 90 degrees Celsius, and it was rich in hydrogen gas produced by a chemical reaction between seawater and the rocks below. To the researchers who study the origin of life, Lost City was almost too good to be true. Because those alkaline vents, with their warm temperatures, their hydrogen-rich fluids and their intricate mineral structures, provided something extraordinary. A natural proton gradient. You may not be familiar with that term, but it is arguably the most important chemical process in the history of life on Earth. Here is the concept. Every living cell on Earth, every bacterium, every plant cell, every cell in your body, generates energy using a process that involves pumping protons across a membrane. This process, called chemiosmosis, was discovered by British biochemist Peter Mitchell in the 1960s. A discovery so unexpected and so counterintuitive that it took the scientific community nearly 2 decades to accept it, and eventually earned Mitchell the Nobel Prize. The key insight is this: life doesn't just use chemical reactions to generate energy. It uses gradients, differences in concentration or electrical charge across a membrane, to do work. Your mitochondria right now are essentially tiny turbines using the flow of protons across their inner membrane to synthesize the molecule ATP, which is the universal energy currency of all living things. The astonishing thing about alkaline hydrothermal vents like Lost City is that they naturally generate exactly this kind of proton gradient between the alkaline vent fluid and the more acidic ancient ocean. No membrane required, no prior biochemistry necessary. The gradient is there, built into the geology of the system as a free gift from physics and chemistry. The researcher who has most powerfully developed this idea is Nick Lane, a biochemist at University College London, whose work in the last 2 decades has fundamentally shifted how many scientists think about the origin of life. Lane's argument, built on decades of careful analysis of the biochemistry of the simplest living organisms, is that the first proto-life did not need to invent the proton gradient. It simply needed to exploit one that already existed in the mineral-lined micropores of an alkaline hydrothermal vent. Picture those micropores. Tiny chambers, a fraction of a millimeter across, lined with iron and sulfur minerals that can catalyze chemical reactions. Warm fluid flowing through them, carrying hydrogen and carbon dioxide. A natural proton gradient driving chemistry forward. And, crucially, thin mineral walls that could serve as a primitive substitute for the membrane that all true cells eventually evolved. In those conditions, Lane and his colleagues argue, the basic chemistry of life, the reactions that fix carbon dioxide into organic molecules, that generate the precursors of amino acids and nucleotides, could have happened spontaneously. Not because some magical ingredient was present, but because the thermodynamics of the system made those reactions not just possible, but, in a real sense, inevitable.
But not everyone agreed that the deep sea was where the story began. And some of the most provocative ideas about the origin of life came from completely unexpected directions. In the 1980s, a Scottish chemist named A.G. Cairns Smith proposed something that, on first hearing, sounds almost absurd. He suggested that the first self-replicating systems on Earth were not organic molecules at all. They were clay. The microscopic crystals that form the layered structure of clay minerals can grow, incorporate environmental chemicals, and produce daughter crystals that inherit the parent's structural patterns.
Cairns Smith called this genetic takeover. The idea that mineral crystals were the original replicators and that organic chemistry gradually assumed their informational role. The way a building's scaffolding is removed once the structure can stand on its own. The clay hypothesis never became mainstream, but it did something important. It forced researchers to think far more broadly about where self-replication could begin and what it might look like. It opened the conceptual landscape. Around the same time, a German lawyer turned chemist named Gunther Wächtershäuser proposed that the first metabolism occurred not in a primordial soup, but on the surface of iron pyrite crystals, fool's gold. Pyrite surfaces can drive the reduction of carbon dioxide into organic molecules, and Wächtershäuser argued that life began not as a free-floating replicating molecule, but as a chemistry anchored to a mineral surface. His specific model remains contested, but the iron-sulfur clusters he highlighted are real. They appear at the heart of some of the most ancient proteins in all of biology, including those that perform the first steps of metabolism in modern cells. He was pointing at something genuine, even if the full picture was more complex than he imagined. And then there was the most audacious proposal of all. In 1973, Francis Crick, who had co-discovered the structure of DNA, and the astronomer Fred Hoyle independently proposed what came to be known as panspermia, the hypothesis that life on Earth did not originate on Earth, but arrived from space, carried dormant inside comets or meteorites. Crick went further, suggesting what he called directed panspermia, that life was deliberately seeded here by an advanced civilization elsewhere in the galaxy. He was not joking. He considered it a serious scientific hypothesis. The mainstream response has always been the same. Even if panspermia were true, it doesn't solve the problem. It moves it. Life still had to originate somewhere, by some chemical process. But the discovery of amino acids on comets, of complex organic molecules in interstellar clouds, of microorganisms surviving simulated spaceflight in laboratory experiments. None of this makes the cosmic dispersal of life impossible. It remains, in modified form, a live hypothesis. Not as an alternative to the chemistry of origins, but as a complement. Life's raw materials may be truly cosmic, even if the specific moment of ignition was local. This is where the story takes its most extraordinary turn. Because even if we grant everything we have discussed, the amino acids from space, the warm alkaline vents, the mineral scaffolding, the chemical gradients, we still face the central problem. How does chemistry become information? How do you go from reactions to a code? The answer came from a molecule that most people have heard of, but few truly appreciate. a molecule that may be the single most important object in the entire history of life on Earth. That molecule is RNA. You probably know its more famous cousin, DNA, deoxyribonucleic acid, the double helix, the molecule that carries the genetic code. DNA is the archive. It stores the instructions. But DNA by itself cannot do anything. It cannot catalyze chemical reactions. It cannot build proteins. It cannot replicate itself without help. DNA is inert without a supporting cast. The supporting cast is made of proteins. Long chains of amino acids folded into precise three-dimensional shapes that perform an astonishing variety of chemical tasks. Proteins are the workhorses of the cell. But proteins are built according to instructions stored in DNA. And DNA is copied and maintained by proteins. Each depends on the other. You cannot have one without the other. It is the most fundamental chicken and egg problem in biology, and for most of the 20th century it seemed to represent an insurmountable barrier to understanding the origin of life.
RNA, it turned out, can do something that neither DNA nor proteins alone can do. It can store information, like DNA, and it can catalyze chemical reactions, like proteins. It is, in other words, a molecule that can potentially do both jobs at once. The discovery of catalytic RNA, molecules called ribozymes, by Thomas Cech and Sidney Altman in the early 1980s earned them the Nobel Prize in Chemistry in 1989. And it opened a door that transformed our understanding of the origin of life. If RNA can both carry information and perform catalysis, then perhaps the very first living systems did not need DNA or proteins at all. Perhaps life began in an RNA world. A world where RNA molecules, forming spontaneously from simpler precursors in the warm chemistry of the early Earth, stumbled upon the ability to copy themselves. And once a molecule can copy itself, everything changes. Think about what self-replication means. If you have a molecule that can make copies of itself, and those copies are slightly imperfect, as copies almost inevitably are, then you have variation. If some variants are better at copying themselves than others, you have selection. And if you have variation and selection operating over long periods of time, you have evolution. Not biological evolution, not yet. Chemical evolution. Molecular Darwinism. The survival of the fittest molecule. In 1967, a biophysicist named Sol Spiegelman demonstrated something that stopped his colleagues cold. He took a simple RNA molecule capable of self-replication and placed it in a test tube with the necessary raw materials, but without any living cell to guide the process. And he watched what happened. Over the course of hours, the RNA molecules competed, mutated, and were selected for the fastest replication. The slower variants were outcompeted and disappeared. What remained was a stripped-down, ruthlessly efficient replicator. A molecule that had discarded everything except its core ability to copy itself. Spiegelman's colleagues named the result Spiegelman's monster. It was the first direct observation of Darwinian evolution operating at the molecular level, entirely outside any living organism. And it demonstrated something deeply unsettling.
that evolution does not need life. It needs only information, replication, and error. The machinery of cells, the membranes, the proteins, the elaborate molecular infrastructure is almost incidental to the underlying logic. Evolution is older and deeper than biology. It is a mathematical inevitability, playing itself out in whatever molecular medium is available. This is the conceptual breakthrough at the heart of modern origin of life research. The moment a molecule could copy itself, even imperfectly, even clumsily, even at an agonizingly slow rate, the ratchet of evolution began to turn. And once it began to turn, there was no stopping it. But the RNA world hypothesis, compelling as it is, has its own problems. Chief among them, how do you get RNA in the first place? RNA is not a simple molecule. It is a polymer, a long chain of smaller units called nucleotides. Each nucleotide consists of 3 parts: a sugar molecule called ribose, a phosphate group, and one of 4 nitrogen-containing bases: adenine, guanine, cytosine, or uracil. Assembling these components into a functional RNA molecule under the chaotic conditions of the early Earth without any biological machinery to guide the process is a chemical challenge of formidable difficulty. For decades, this was one of the most serious objections to the RNA world hypothesis. Chemists knew that RNA was capable of the right tricks, but could not figure out how it might have assembled itself from scratch. The individual components were hard to synthesize under plausible prebiotic conditions.
Getting them to link together in the right order seemed to require conditions that were mutually exclusive. Then, in 2009, a chemist named John Sutherland at the Medical Research Council Laboratory of Molecular Biology in Cambridge published a paper that sent shockwaves through the field. Sutherland and his colleagues had found a new route to RNA nucleotides, not by assembling the pieces separately and joining them, but by starting from simpler precursor molecules and following a chemical pathway that produced nucleotides directly. When they mixed cyanamide, cyanoacetylene, glycolaldehyde, glyceraldehyde, and inorganic phosphate, all molecules that would have been available on early Earth, and subjected them to wet-dry cycles and ultraviolet light, they got RNA nucleotides. Good ones. The kind that could link into RNA chains. But what Sutherland's team noticed next was even more striking. The same chemical conditions that produced RNA nucleotides also produced the precursors of amino acids and the precursors of the lipids that form cell membranes. All 3 classes of molecules at once, from the same starting materials, under the same conditions. This was not what anyone expected. The standard assumption had always been that the different components of the cell arose separately and then somehow found each other. Sutherland's chemistry suggested something more radical, that they co-emerged, that the chemistry of life is not 3 separate problems that happened to be solved independently. It is one integrated chemical network that produces all its components simultaneously, as naturally as a river produces both water and sound. And there is one more puzzle in this system that deserves its own moment. A puzzle so strange and so specific and so unexplained that it has haunted chemists for decades. It is the problem of chirality, the problem of handedness. Most organic molecules can exist in two forms: mirror images of each other, like a left hand and a right hand. Chemically, they have the same composition, but they are not interchangeable. They interact differently with other molecules, the way a left glove fits only a left hand. Here is the extraordinary fact. Every single amino acid in every single protein in every living organism on Earth is left-handed. Every sugar in DNA and RNA is right-handed. Without exception, everywhere, in every bacterium in the deepest ocean trench and every cell in the canopy of the highest rainforest. Life chose a handedness, and in 4 billion years it has never once deviated from that choice. But chemistry, operating without guidance, produces left-handed and right-handed molecules in equal numbers. There is no obvious chemical reason why life should prefer one orientation over the other, and yet it does, with a consistency that is absolute and universal. Where did this preference come from? We don't fully know. Some researchers point to the slight asymmetry built into the fundamental laws of physics, an asymmetry so tiny it seems almost too small to matter, yet perhaps sufficient to tip the scales in a world of fierce molecular competition. Others suggest that the first self-replicating RNA happened, by chance, to be one-handed, and that handedness was enforced and amplified by selection. Some look to polarized light from nearby neutron stars, which can selectively destroy molecules of one orientation and leave the other intact. What we know is this: life made a choice. It made that choice once, at the very beginning, and it has been kept faithfully ever since through every generation of every organism that has ever existed. The handedness of the amino acids in your proteins connects you, directly and without interruption, to a molecule that formed in warm water 4 billion years ago.
That single choice, preserved through all of evolutionary time, is perhaps the most intimate reminder of how deep your roots actually go. We are now approaching the threshold of the first true cell. We have molecules, we have RNA capable of self-replication, we have chemical gradients providing energy, we have membranes assembling themselves spontaneously from lipids. The question is no longer whether each component could form, it is whether they could find each other, and what happened when they did. Jack Szostak, a Nobel Prize-winning biochemist at Harvard, has spent much of the last 2 decades studying exactly this question. His laboratory has shown that fatty acid vesicles, simple lipid bubbles, can grow, divide, and encapsulate RNA in ways entirely consistent with the chemistry of the early Earth. When RNA is enclosed inside a fatty acid vesicle, something remarkable happens. The RNA creates an osmotic pressure that actually drives the growth of the membrane, pulling in new lipid molecules from the surrounding solution, making the vesicle larger, more robust, more competitive. The genetic material and the cellular container help each other. They are not 2 separate problems that had to be solved and then assembled. They solve each other simultaneously from the moment they come into contact. This mutually reinforcing chemistry is perhaps the deepest reason to believe that the transition from molecules to cells
was not a single improbable leap, but a continuous self-driving process. Each improvement in replication gave a slight advantage. Each advantage was inherited by daughter vesicles at the next division. The ratchet turned notch by notch over millions of years. And what emerged at the end of that process was something the early Earth had never contained before: a cell.
A bounded, self-sustaining, self-replicating chemical system, maintaining a sharp distinction between itself and the outside world, reading a genetic code, building proteins, harvesting energy, and passing all of this on to its offspring with extraordinary fidelity. The first true unit of life. Is this life? The question matters more than it might seem, because the boundary between a sophisticated protocell and a true living cell is not a sharp line. It is a gradient, a long slope of increasing complexity and integration along which, at some point, we agree to plant our flag and say, here is where chemistry became biology. But wherever we plant that flag, one thing is clear. The process that led to it was not magical. It was not directed by any external intelligence. It was not the product of an improbable cosmic coincidence. It was the product of chemistry, following its own logic, in environments that the early Earth provided in abundance. The emergence of life was not an exception to the laws of physics and chemistry. It was an expression of them. Scientists call the ancestor of all modern life Luca, the last universal common ancestor. Not the first life, that was probably far simpler, far more primitive. But the last common ancestor of every living thing alive on Earth today. The single population of organisms from which every bacterium, every fungus, every plant, every animal ultimately descends. What was Luca like? This is a question that can actually be investigated scientifically by comparing the genomes of modern organisms and working backward to reconstruct what genes must have been present in their common ancestor. The results, published in a landmark paper in 2016 by the German biochemist William Martin and his colleagues, were striking. Luca, according to this analysis, was not a simple thing. It already had hundreds of genes. It was already adapted to a specific environment. The analysis pointed strongly toward a hydrothermal vent environment, hot and hydrogen-rich. It already had the basic molecular machinery for protein synthesis, including a primitive ribosome, that remarkable molecular machine that translates the genetic code. It was already, in other words, a sophisticated living system. Which means that by the time Luca existed, the hardest part of the origin of life, the transition from chemistry to the first coded protein synthesizing cell, was already complete. Luca was not at the beginning of life. It was already, in a real sense, well past the beginning. Where was the beginning? We don't know. We may never know. The geological record from the first 500 million years of Earth's history is almost entirely erased, destroyed by the violence of the late heavy bombardment, by the heat of the early mantle, by 4 billion years of geological recycling. The very rocks that might have preserved the earliest chemical traces of life
have long since been subducted into the mantle or metamorphosed beyond recognition. What we have instead are hints, whispers from the deep past. Carbon isotope ratios in ancient rocks from Greenland, dated to 3.7 billion years, that are consistent with biological activity. Putative microfossils in Australian chert, dated to 3.5 billion years, though their biological origin has been debated. Chemical signatures in ancient minerals that some researchers interpret as evidence of life, though others remain skeptical. The honest answer is that we don't know exactly when life began on Earth. We don't know exactly where. We don't know exactly how. We have fragments of evidence, powerful theoretical frameworks, and increasingly sophisticated laboratory experiments that shed light on the plausibility of various pathways. But the actual origin of life, the specific sequence of chemical events that led from dead matter to the first living cell, remains, for now, beyond our direct reach. And yet, the story we've told tonight, from Miller and Urey to the Black Smokers to Spiegelman's monster to Shostak's vesicles, is not a story of failure. It is a story of a species slowly, patiently, molecule by molecule, learning to read one of the deepest chapters ever written. We have not finished reading it, but we can see the shape of the argument. We understand the logic. And for the first time in human history, we are building in the laboratory the very systems we believe gave rise to life. Testing our understanding not by observation alone, but by creation. That understanding, when it finally becomes complete, will not diminish life. It will not make the living world seem mechanical or cheap. Just the opposite. If life is a natural consequence of chemistry, if, given the right conditions, the emergence of self-replicating, evolving, thinking matter is something the universe does as readily as it forms stars, then every living thing is not a lucky accident. It is the universe knowing itself. And it all began on a poisoned planet, in warm, dark water, with a molecule that learned, for the first time, to make a copy of itself. We started tonight with a world without life, a world of rock and toxic gas and silence. And we've arrived, through the astonishing alchemy of chemistry and time, at the threshold of everything that follows. The bacteria that will learn to breathe, the cells that will learn to cooperate, the animals that will crawl from the sea, the forests, the dinosaurs, the mammals, the primates. The moment, perhaps 60,000 years ago, when a human being looked up at the night sky and wondered, for the first time, what it all meant. All of that, every chapter of that story, begins here. In the warm, dark, chemically restless waters of an early Earth that had no idea what it was about to become. Thank you for spending this time with us tonight. If this kind of story moves you, if the deep history of life on Earth is something you want to understand, then this channel is for you. There is much more to come. Next time, we go one step further, from the first molecules to the first true cells, from chemistry to biology, from the RNA world to the world of DNA and protein. The story is just beginning. Sleep well, and remember, the atoms in your body have been on an extraordinary journey
Tonight, for a little while, they got to hear about it.